feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yong Lin School of Medicine's Healthy Longevity webinar. We have a really exciting seminar tonight. It's Andrew Teschendorff will be joining us and talking about epigenetic aging clocks and where they're going in the future. And for that, please use the Q&A function and ask questions, and we'll get to as many of those as possible. Before we do that, however, we have Teresa Liao, a research associate at the NUS, and she will talk about her own study, which involves implications of post-exercise human serum.
Thank you for introducing me. As mentioned, my name is Teresa, a research staff member at the Healthy Longevity Program. Today, I'm excited to share some ideas about the potential implications of human post-exercise serum. We all know that exercise is good for our health, but how exactly does it work? When is the right time to start exercising? And what if someone hasn't been active for years? Is it too late to start now? This slide shows data from a 35-year longitudinal study examining how mortality changes with different levels of physical activity. The green line represents people who increase their activity intensity from low or medium to high. The brown line represents those who maintain high activity levels throughout. Interestingly, men who increased their activity levels between ages 50 and 60 initially had a higher mortality rate in the first 5 years, but after 10 years of follow-up, their mortality dropped to the same levels as those who had always been highly active. This shows that it's never too late. Even starting intense physical activity later in life can still lead to significant health benefits over time. But what's happening inside the body to drive these benefits? Let's look at one of the key mechanisms, the inflammatory response to exercise. Exercise triggers a controlled inflammatory response, and that's an actually good thing. After a single bout of exercise, the first phase of inflammation involves phagocytosis, where immune cells clear out damaged tissue. This is followed by a regenerative phase marked by central nucleation, a sign of muscle repair and growth. During this whole process, the body releases various cytokines like interleukin-6, TNF-alpha, IL-1 beta, and irisin, along with stems such as HNGB1 and heat-shock proteins. These molecules help signal tissue repair, adaptation, and even systemic health benefits. So we've seen that exercise triggers powerful biological processes that support longevity and muscle health. However, not everyone is able to exercise, whether due to age, injury, chronic disease, or limited mobility. This raises an important question. Can we find alternative ways to deliver the benefits of exercise to those who can be physically active? To explore this idea, we come up with a post-exercise serum control media. We collect blood samples from healthy individuals after exercise. From this, isolate the serum, which contains various exercise-induced molecules like cytokines, myokines, and DAMs. We then use this post-exercise serum as the culture media for C2C12 cells. The goal is that the bioactive factors in the serum will stimulate cellular pathway in these muscle cells, promoting changes in the gene and protein expression related to muscle regeneration, growth, and inflammation resolution, similar to what happens in the human body during recovery and adaptation after exercise. This study provides evidence supporting our concept. They collected blood from individuals who had been engaging in long-term exercise such as swimming, bodybuilding, soccer, and volleyball, and used their serum as culture media in human muscle cell line. The results show that these long-term exercisers had a significant highly myotube fusion index compared to those with untrained blood. Building on the concept we have just discussed, our study takes a unique step forward. To our knowledge, no one has yet systematically explored how human post-exercise serum stratified by both age and exercise type can influence muscle cell behavior in vitro. Our study model aims to investigate how exercise-induced myokines and DAMs contribute to muscle health and longevity. We will analyze the protein and gene expression of exercise-induced myokines and DAMs to understand their role in muscle repair, growth, and adaptation. This will help us explore whether we can mimic the beneficial effects of exercise at the cellular level, even for those who may be unable to engage in physical activity themselves. Thank you for your listening.
Thank you. It's interesting to see the effects of starting exercise later in life. Can be good or bad, I guess. Have to look into that. I want to introduce Professor Andrew Teschendorff, who is the principal investigator at the Shanghai Institute of Nutrition and Health, part of the Chinese Academy of Sciences, where he leads a laboratory in computational systems epigenomics. He was recognized as a Clarivate highly cited researcher in 2023 for his work on epigenomics, aging, and cancer systems biology. Thanks for joining us, Andrew.
Well, thanks, Brian, for the very kind invitation. I'm very happy to, delighted to be part of this very prestigious webinar series. So thank you so much. So the topic of my talk is going to be on epigenetic aging clocks. I talked about this 2 months ago in Singapore as part of the geophysics meeting. But today I'm going to present a lot of new material, I hope. So some of it is recently published, very recently published. Some of it is under review, and the other last part is about to be submitted. Okay, so I will give a brief introduction to DNA methylation clocks, and then much of the discussion today is going to be focused on the effect of cell type heterogeneity and how this affects the interpretability of epigenetic clocks, and also some of the how this ties in with some of the work we've done also in relation to inflammation. And then the last part of the talk is going to be about a new approach that we are proposing to classify the diversity of age-related DNA methylation patterns, which I think is a challenge that has been overlooked in the epigenetic clock field, and I will explain why. Okay, so let me start with a definition. For me, a molecular aging clock is a counter that keeps track of age cumulative molecular alterations as measured relative to a suitably defined ground state, and that can quantify either chronological age or various forms of biological age, which may include all-cause mortality, disease risk, or various forms of cellular age, so for instance, telomere length or mitotic age. And as I'm sure you all know, DNA methylation clocks were the first clocks to be proposed. It's worth remembering, though, that at least in the cancer context, somatic mutational signatures have been were characterized a long time ago as well. And some of these signatures also correlate with chronological age. And the link between somatic mutations and DNA methylation clocks was recently explored in a very nice paper by Trey Adeka's lab. And more recently, we've also seen many studies proposing transcriptomic, proteomic clocks, which seem to be very promising as well, and metabolomic and histone mark clocks. So the focus of today's talk is on DNA methylation clocks. There are a number of reasons why DNA methylation clocks stand out. So one is that they are probably the most accurate for chronological age prediction. So some of the recent proteomic clock papers also claim very high accuracies, but it's worth remembering that the DNA methylation clocks, like the Zhang or the Horvath clock, have been validated across 50 or more independent DNA methylation data sets. That level of stringency and validation is still waiting. In the case of the proteomic clocks, they've only been validated across 2 or 3 independent data sets. Another important reason, or what makes DNA methylation clocks interesting, is the fact that they work across different species. So now we have technologies as well that allow us to measure methylation patterns in mice and other mammalian species. So this is something that is really important, especially in relation to the fact that these methylation clocks have also been shown to predict cellular rejuvenation upon age reversal interventions in animal models. And last but not least, the DNA methylation clocks have been validated also in relation to different quantifiable forms of biological age. So not just all-cause mortality from a blood sample, but also cancer risk from actual solid tissue. So for instance, cervical smears. Okay, so one of the big challenges, though, with epigenetic clocks is interpretability. We know that there are composites of many different underlying processes. And one of these processes, of course, is cell type composition, which changes with age. So 2 years ago, we published this meta-analysis where we used the 12 immune cell type reference panel to estimate fractions for these 12 immune cell types across 20 different cohorts, so different DNA methylation data sets. And as you can see, and as expected, we find this consistent decrease in the naive CD4 and CD8 fractions. But also, there were other patterns that were fairly consistent. For instance, the increase in the basophil fraction or the increase in the natural killer cell fractions. And so some of these findings have been validated. We've been able to validate them using single-cell RNA sequencing studies, which have been generated on close to 1,000 individuals. What is also important to note is that these fractions also correlate with health outcomes. So this was done in collaboration with Jessica Lasky-Sue from the Harvard University, so using the Mass General Biobank data. You can see how the naive CD4 T cell fraction is protective for all-cause mortality to a degree which is actually very similar to that of the epigenetic clocks, raising questions as to whether the associations that we observe for epigenetic clocks, whether these are also independent of these variations in cell type fractions. Okay, so before I address that question, let me address the simpler question, which is how much of the accuracy of a clock is due to changes in cell type composition. So this is reflected here by, or described here by this, what I call extrinsic process, and how much of it is due to intrinsic changes in individual cell types. So this can be addressed as follows. So if you take, let's say, one of these data sets, a blood data set with about, these are 700 samples, you can then build 2 clocks. You can build a clock unadjusted for the cell type heterogeneity, and then you can build another one which is adjusted for that cell type heterogeneity. And we did it at 2 resolutions, at the resolution of 12 immune cell types, and at a lower resolution where we bunched together memory and naive subsets for each of the T cell categories. And this is the average result across the studies. You can see that about 40% of the accuracy of a clock is driven by an extrinsic process, so variations in the immune cell type composition. Around 20% of that is due to shifts in the naive and memory T cell subsets. This extrinsic fraction actually drops quite a lot in the case of brain. This is probably an underestimate because with brain tissue, we don't have the ability to characterize the heterogeneity as reliably as with blood, and also not at the higher resolution. So this was done at the resolution of 7 brain cell subtypes. Okay, so how about the impact of cell type heterogeneity on age acceleration measures from these clocks, and also in relation to health outcomes? To address this question, you would want to build a reference panel at a much higher resolution, not just 12 immune cell types, but to include also cell types which characterize the heterogeneity within the innate immune system. And this is a very challenging problem. What I'm going to describe to you now is really a spin-off from another project that I've done in collaboration with Charlotte Cecil and Bas Heijmans from the Netherlands. They were interested in building a unified DNA methylation reference panel that would work for a blood sample of any age, so including birth, babies, toddlers, children, up to adulthood. And to build this reference, we took the following strategy. So we took sorted cell types from cord blood and sorted cells from adults. And we then merged the data and performed the PCA analysis. And this PCA analysis was very revealing. So PC1 correlates with the myeloid and lymphoid cell discrimination, which is what you would expect. But PC2 showed associations simultaneously with antigen exposure. So these are the adult naive CD4, CD8 T cells. These are the memory counterparts, but also was able to distinguish the cord blood, which are also antigen inexperienced T cells, from the adult naive counterparts. So this is sort of an aging effect. So based on this, we then hypothesized that if we have, in this reference, we have a model or a methylation profile for youthful monocytes and adult monocytes, that maybe some of the age accelerations we're measuring with these clocks, that this is not just due to changes in the T cell compartment, but also due to shifts in the innate immune system. So to check this, we performed a random effects meta-analysis over 15 cohorts. So we looked at 4 of the well-known clocks, Horvath, PhenoAge, GrimAge2, Dunnet and Pace. And as you can see, there are the expected associations. So for instance, if you take Dunnet and Pace, which is probably the one that is most strongly associated with biological age, you can see that the naive CD8 and CD4 fractions are anti-correlating, so they're decelerating the clock. But you can also see how the adult monocyte fraction is correlating positively and how the youthful monocyte fraction is actually correlating negatively, suggesting that indeed there is heterogeneity within the innate immune cells, which is contributing to these age acceleration measures. Now, to anchor this on biology, we hypothesized that maybe some of this variation is related to inflammaging. So what we did was to take the CPGs that were identified from a multi-ethnic EWAS for serum CRP levels and to build an inflammation score. So this is derived from DNA methylation data, and think of it as a proxy for CRP serum levels. So it correlates with age as it ought to. It's increased in inflammatory conditions. And as expected, this inflammation score correlates very nicely with the age acceleration measures of these clocks. I mean, this is not surprising in a way, because CRP was one of the clinical biomarkers that was used in the PhenoAge, and is also one of the 16 or 19 biomarkers that were used in the Dunnet and Pace clock. This is another clock that was published this year from Skin and Comboy's lab. This one does not correlate with CRP levels, and we understand why. This is actually one of the reasons why you need to be very careful about cell type heterogeneity, because they didn't really adjust for the cell type compositional changes, and this is clearly measuring something very different. So how do these fractions now correlate with the Inflammaging score? This is the result for the older reference panel. So you can see how the naive and the memory T cell fractions correlate in the expected direction. If we take the total monocyte fraction, that is not correlating with this with this Inflammaging score. However, if we take the higher resolution reference panel, now we do observe a correlation. So the adult monocyte fraction is correlating positively. The more youthful monocyte fraction is correlating negatively. Incidentally, if we were to subtype monocytes in a different way, so if we were to subdivide them by the classical and non-classical subtypes, under a random effects model, which is the right model to consider because there's substantial heterogeneity, as you can see, we do not see a consistent pattern, suggesting that this way to subdivide monocytes may be actually very, maybe biologically relevant and maybe something that is worth to pursue, investigate further. How do these fractions correlate with health outcomes? So we teamed up with Riccardo Marioni and investigated this in the Generation Scotland cohort. So these fractions, so for instance, the monocyte fraction correlates with various diseases, notably liver diseases, but also chronic obstructive pulmonary disease. And we also did an analysis in relation to all-cause mortality. So again, validating the previous results, the naive CD4 T cell fraction is the one that is most predictive and it's protective of all-cause mortality. But this other monocyte fraction is also correlating and the youthful monocyte fraction is marginally protective of all-cause mortality. And so what is the implication of this for the age acceleration measures of the clocks? So if we adjust for the 19 immune cell fractions, we can see that, for instance, the Dunedin pace, the pheno-age clocks still remain associated with all-cause mortality. Although there's a drop in the association, as you might expect, it's still independent of it. So this is consistent with the view that we have a very strong intrinsic component to these clocks. Okay, so it's also of interest, of course, to dissect the intrinsic clocks, so the changes happening in individual cell types in terms of building cell type specific clocks. And this is something that I talked about 2 months ago. So I think probably in the interest of time, I will just fly through this because it's already on YouTube. So just to make you aware that we have been developing methods that allow us to estimate cell type fractions in many different tissues. And once you estimate these fractions, you can then use statistical methods to infer in which particular cell types the methylation changes are happening in. A caveat to this, though, is that you need a large number of samples, okay? Which for bulk tissue, that may well be the case that you have access to such large data sets. Certainly, that's very difficult for sorting cells or prohibitive in the case of single cell methylation data. Okay, so we, as a proof of principle, we showed that we can build hepatocyte clocks. We showed that it can predict biological age acceleration in various conditions. And we also showed this in the context of brain, building neuron and glia-specific clocks, showing that they were specific to brain, so they predict age better in brain than in other tissues, and also performing a meta-analysis in Alzheimer's disease, showing that the glia clocks in particular were strongly accelerated, especially in the temporal lobe. And these clocks were also showing significant overlap with the damaged causal clock from Vadim Dadashev's lab. And what was actually quite striking was that these small number of CPGs were actually mapping to genes that have been implicated in Alzheimer's disease, suggesting that this cell type specific approach is pointing in the right direction. Now, I'll come now to the last part of the talk. And this is our effort to define a new taxonomy of the aging methylome. So in order to understand the motivation for this, remember that epigenetic clocks, when you build them, you need to make a choice of a machine learning algorithm and model, as well as an outcome variable. And what perhaps has not been sufficiently appreciated in the community is that when you make this choice of algorithm, this is imposing significant constraints on the types of features that you select. So this is a very nice figure from my collaborators in Australia near Enon's lab. And what this shows here is 3 different patterns of age-related change that we observe, right? So there are these CPGs that change linearly with age, but where the variance is fairly constant. Then you have CPGs that change linearly with age, not perhaps with such a big gradient. They tend to have smaller gradients, but where there's an increase in variability. And then you have these CPGs where the variance is, the methylation, the mean level is constant, but there's an increase in variability. And if you use traditional machine learning methods, and by the way, by that I also include neural networks, we tend to select these guys because these are the ones that have less variance, and so they are more predictive. But these guys are also really important. So we actually, over 12 years ago, we showed in the context of cancer, these were pre-neoplastic lesions, prospective collective samples, that those CPGs that show this increased variability are much better predictors of cancer risk, future cancer risk. And this can be understood by the fact that these big changes in methylation, these are capturing subclonal expansions. And these subclones then are the ones that are marking cancer risk. So there's a clear connection there and an understanding why they might be better. So, and these CPGs were recently explored in a paper from Comboy's lab, where they proposed this idea of a noise barometer. Now, the key point to note here is that this is an underrepresentation. It's not a very realistic representation of all the diversity of age-related patterns that you can see in the methylome. And I like to use this analogy. It's more like trying to classify let's say different species. So say you've got an image, and you want the algorithm to tell you which animal it is. And we know that deep convolutional neural networks have been revolutionary for image recognition and classification tasks. So the question we asked is, can we use deep learning to classify age-related CPG patterns? So the key idea here is that we transform the methylation versus age scatter plots into images. And then we use a deep learning method to try to identify the different patterns. As you can see here, for instance, we may have a pattern where you've got a CPG that is very stable, except for a small number of outliers. Or you may have a CPG profile that is bimodal, but where only one of the modes shows a change with age. Could be a sex-specific DNA methylation change. So we tried many different approaches. We tried unsupervised methods. We teamed up with a group in the US who are experts on deep learning. We also teamed up with a group in China who are experts on deep learning. But the unsupervised approach sadly failed. So I'm really keen to hear from anyone who may have better ideas. So what we had to do was to revert to semi-supervised approach, where we use statistical testing and also distributional properties like kurtosis, which is related to the 4th order moment of a distribution, to define the CPG categories. And then we train the deep learning model. This is then validated on independent CPGs. And we then use this to classify the rest of the CPGs. And so by using this approach, we can, this was done on a large EPIC data set of 3,500 samples. So it's about 850,000 CPGs. So approximately 70% of these CPGs can be classified this way. So here you've got representative examples of the different categories. So for instance, here you've got a trimodal CPG. This is an MQTL, where the change with age is only happening in one of the genotypes, okay? Then you've got this unistable one, where the methylation is just beautifully stable, as you can see. And then you've got these uni-unstable ones where, again, it's unimodal, no change with age, but it's more variable than the unistable one. So the algorithm works reasonably well in terms of validation accuracy. And does it make sense, this classification? So one way to test this is to use a tool like eForge2 to test for enrichment for chromatin states. We also looked at correlations with immune cell fractions. And these results make sense. So for instance, lineage homo, these are the linearly related CPGs with constant variance. these are strongly enriched for enhancers. And they also show strong correlations with the immune cell fractions. So this makes sense because the cell type specific CPGs are enriched for enhancer regions. And so these are the ones that are really capturing the variations in immune cell fractions. But there were also some really big surprises. So the lineage het category, these are the ones that show this increase in heterogeneity. So the methylation increases or decreases with age, but there's also increased variability. These are actually strongly associated with variations in the memory CD8 T cell fraction. This was totally unexpected. The unistable ones, they are enriched for active promoters. So here you need to make a distinction between those that are lowly methylated, and the ones that are highly methylated. So these map to the gene body, as shown by this enrichment for the K36 trimethylation mark, whereas the lowly methylated ones are mapping to the transcription to the promoters. So these are basically genes that are highly expressed in these tissues, and they have to be. We also looked at the enrichment for for epigenetic clocks, and there were some interesting new insights that came out here as well. So, for instance, this lineage het category, which are these CPGs where you get this increased variability, these were actually most strongly enriched for mitotic clock CPGs. So again, I think the interpretation here is that what you're seeing is clonal hematopoiesis, because remember, these were derived from blood. And these subclones, or this clonal hematopoiesis is reflected by the increased cell proliferation, which is being captured by these mitotic clocks. The tri-H category, these are mostly MQTLs, and consistent with that, you see the very strong enrichment for the causality clocks, right, which were built from the outset using MQTLs and then correlating the methylation to health outcomes. Now, this other category, these are the ones where the level of methylation stays constant, the average level, but where there's an increase in variability. These are generally speaking not enriched for any of the clocks except this one. This is one that we've just built. So, and we built it on purpose to try to explore this CPG category further. So you can see that there's a little bit of enrichment for the Dunedin pace and PhenoAge, but otherwise it's really depleted of all these different clocks. So we decided to explore this further, following the footsteps of Conboy's lab. But we decided to use more cohorts. They only looked at one cohort. That's not enough. So we took 3 large cohorts, different ancestry. We adjust for the variations in the immune cell type fractions because that is a driver of this variability. And we derived 493 CPGs that we called noise clock CPGs. These are, generally speaking, weakly correlated, but technically it's not noise because it's still more correlated than randomly selected CPGs. But certainly they do not display strong correlations. And so from this, you can then build a measure of variability or noise for each sample. And this correlates with inflammation, as shown here, although the association with age is stronger by construction. So by construction, it correlates much more strongly with age. So if we add the set type fractions as covariates, then you still get this association with inflammation. So in other words, these non-H het CPGs, they are correlating with inflammation as measured by CRP levels in a manner that is also independent of the variations in immune cell fraction. So that's actually quite important to establish that. This is the last slide because I'm sure that I'm already running over, as I usually always do. So, so this is a work in progress, I should add, where we are trying to understand these genotype-specific HMQTLs. So these are trimodal CPGs where there's an association with age, but only in specific genotypes. So in this case, it's just the BB genotype. So unlike the broad category of HMQTLs, which are strongly enriched for the DAM-H and ADAPT-H course of CPGs, these genotype-specific ones are depleted, or they're not really present in these clocks. And yet they are important, as we show here. So a lot of the variation that we see as a function of age, this is related to inflammation. At least we think it is, because it's correlating with CRP levels, also with IL-6 levels, and also various episcores not shown on this slide. And the associated SNPs are also enriched for inflammatory diseases. So I think that this is a way now to build a new or a separate category of causal clock CPGs, where the association with age and inflammation is really genotype specific. And that could be really interesting, I think. Okay, so these are the take-home messages. So I've shown that in blood, cell type composition can account for 40% of an epigenetic clock's accuracy. In brain, this fraction drops to 15%. With large enough sample sizes, it is possible to build cell type specific epigenetic clocks. The age acceleration of first and second generation clocks is associated not only with shifts from naive to memory T cell fractions, but also with shifts within the innate immune system, so monocytes and natural killer cells predominantly. The clocks, current epigenetic clocks do correlate, display strong associations with CRP inflammation measure. This was derived by independent means using this multi-ethnic EWA study. These clocks also, the associations with all-cause mortality do seem to be independent of all major risk factors, including now these 19 immune cell type fractions. So this is sort of now demonstrating this at a much higher resolution. And finally, I've shown you our efforts to build a taxonomy of age-related DNA methylation patterns. I think this can be a very useful way in which to now use this as an anchor point to then build epigenetic clocks that are sort of looking for specific shapes in the human methylome. Okay, so many people to thank. There's a lot of the work I've shown is done in collaboration with groups with Jessica Lasky-Sue, with Neil Aignan, with Riccardo Marioni, and also a group in Netherlands. Esteban Ballestar, who contributed a lot of the monocyte data. And of course, last but not least, my group. So a lot of the work I've presented has been done by 4 of my students here, Zhao Zhen, Qi Tong, and Xiaolong. And I'm very happy now to take questions. Thank you.
Thanks, Andrew. That's a lot to digest, and I think there's some really interesting data there. You know, it's one of the things I have to say ahead of time is there's 2 types of lecturers that run over time. There are lecturers that know they run over time, and that's what you are. And there are lecturers that run over time every single time and pretend that they don't know they're running over time. And it's the second group that really irritates me, not the first group. So
right.
You're in the right group.
I'm glad to hear that. yeah. Well, you know, I'm yeah, okay. I guess you have to attribute this to my German genes. I mean, Germans like to be very punctual, so if I run over, I feel terrible. I always, but it's just.
So you're not German in the sense that you run over, but you're German in the sense you feel guilty about it.
Yeah, I really feel guilty about running over. yeah. It's just.
So, a lot of things jumped out at me from that, but I thought I would start with a kind of a conjecture which you can shoot down. You know, because when you're talking about aging, you know, you're talking about measuring how a population of humans age. You're also talking about how specific humans age differently than other humans. And it seems to me like a lot of these clock constructions are really focusing on the generalities because that's where the correlation lies, and they, much like a GWAS score, they miss the rare changes or the noise in the system because it's not consistent enough to score. So I'm wondering, you know, when you're sorting things like you do, is there a way to distinguish between measuring aging based on what we all do and measuring aging based on what some of us do?
Yeah, that's a great question. Look, I think that when you think about how these epigenetic clocks have been built, right? So you're going to have, in these large populations, you're going to have, as you say, individuals who are outliers, right? The hope is that somehow these outliers, which could be actually quite frequent, that they are not unidirectional, so that effectively you get outliers, people who are aging more faster than what you would expect for somebody of that age, and also individuals who are aging much which are sort of undergoing healthy aging. So the outliers are good outliers in that sense, and that this is sort of a compensatory mechanism, which means that when you build these clocks from a training set, that it doesn't affect so much the identification of the actual relevant CPGs, right? But it does, of course, raise a major challenge especially in relation to how you may want to apply these clocks in a clinical setting or a translational setting, because there, of course, it's precisely you want to be able to inform the individual as to whether their measurement is reliable, or I mean, is it within what you would healthy aging or not? And currently, we don't have a way or nobody's really thought of a way of quantifying this healthy aging interval, so that you can then more confidently identify those people who are outliers. I mean, that's one part. That's one answer to your question. I guess the other the other way to answer this is precisely in terms of how we select for these CPGs, because maybe training it on chronological age, for instance, is not a good idea, because as we know, it's not necessarily, you're not going to pick out the most relevant CPGs. Right, or for instance, you may want to look for these outlier profiles, which
my worry is that everybody's an outlier in different sites, right? And that's capturing the differences in how we age rather than the similarities, which is what the common sites are capturing. And I'm wondering how to distinguish between those two.
Yes, that's a fantastic point. I mean, this reminds me, in fact, of the work we did in the cancer context, because there, when so let me tell you a little bit about that. So this was a study where we looked at cervical smear samples. These were women that were healthy at the time the samples were taken, but 3 years later, half of the women had developed a cervical intraepithelial neoplasia. And we asked the question, can we actually identify differences in methylation between these two groups? And the answer was that in terms of the mean methylation, you cannot. There was no statistical significance, but it was striking how in those samples from the women who later would develop cancer, you had frequent outliers. So there would be CPGs that were, the methylation was a big change in methylation, 20 to 30% change. And actually, it seemed to be stochastic because they were not shared with the other women who later would develop cancer. So each woman who would develop cancer would have a different set of CPGs. I mean, the overlap would be quite small, very small. And so it's very likely that there's something similar happening in the aging context, that you have these different outliers happening in different individuals, but maybe there's some convergence in terms
I'm wondering how to tease that out. So.
Well, pathways. I mean, you can actually start to map these changes to pathways. I mean, for instance, in the case of cancer, we do see convergence. So even though the CPGs are different, but they are mapping to tissue-specific transcription factors. And so what is happening there probably is that these methylation events are silencing tissue-specific transcription factors, It may be slightly different ones in different individuals, but if you hit 3 or 4 of them, then you get this disruption of differentiation, which usually precedes cancer development. Well, in this case, it's actually the neoplasia. So it may be something similar happening in aging, where you have different CPG sites, maybe even different genes, but they're all mapping to the same pathway.
Yeah. I think that gets back to the challenge with methylation, is that how do you get from methylation sites to mechanism? I mean, that's sort of the benefit of the proteomic or metabolomic clocks, because we're much closer to mechanism there. The challenge is, and the thing that I worry about with these clocks, is the reproducibility from one lab to another running proteomics or metabolomics is very, very low relative to methylation. So, I don't know how any of these clocks are going to survive that sort of variability and noise in the experimental approach. But I think from a mechanistic perspective, they're much better. I guess that gets to the point, which is that, there are multiple uses for these clocks, which is mechanism. How do we understand aging from them? Another one would be, how do we measure traits and behaviors and lifestyles in a population or something like that? Clocks, I think, are pretty good for that. The third one is, how do we measure it in an individual in a clinic? And that's, I think, what a lot of people are struggling with now. So I guess different clocks for different purposes.
Yeah, I agree with everything you've said there. The proteomic clocks, the work that has recently been published, so using the UK Biobank as a resource, that's very exciting. And also there was follow-up work from Vadim Gladishev's lab, right? Building organ-specific clocks. So I think that so I agree with you that the robustness and reliability of these clocks needs to be demonstrated across many more independent data sets. Even with the UK Biobank, with such a large data set, even if you use a training test set strategy, I'm still very concerned, because 20 years of omic data analysis taught me one thing, that sometimes it's not a matter of how many samples you have in a study, because you can have study-specific confounders. And even if you split the data up into training test set, it does not get rid of that. It doesn't get rid of these unknown study-specific confounders. Now, the results from that paper were very consistent and made a lot of sense, so suggesting that there is no major confounder. Although it was interesting that I think 70% of the proteins measured did change with age, 70 to 80%. So that's a very high proportion. That probably explains why it's so accurate. So whether, whether this, want us to see whether if you measure more proteins, what actually happens there. So, and I think that one way to I think an integration of these clocks might be a way to do it. So it would be very exciting to see if some of these proteins that come up from the protein clock analysis and the organ-specific ones, whether this is reflected at the DNA methylation level. Of course, we're not measuring we're not measuring plasma, but but it's. I think there are strategies or avenues of research that could be explored.
Let me ask a more basic question, and then I'll go to the audience. So what are these first generation clocks really measuring? The reason I ask this is because we built a clinical chemistry clock recently with Jan Gruber and Fengsheng, with a little help from me, built this clock using clinical chemistries to predict mortality from the NHANES data set, a historical data set in the US. And recently the methylation data came out. And so we just wondered how different methylation clocks would predict mortality 200 months out from the measurement. And
from what? From 200 what? Sorry, I didn't
months. So 20 years of data. So it collected a cross-sectional data set, and then 20 years more or less later, we looked at mortality data over that period. The second generation methylation clocks did a reasonably good job at predicting mortality, and you might expect that because they were trained on some kind of outcome related to that. But the first generation clocks it was equally good to look at your passport to predict mortality as it was to look at these first generation clocks. And
Well,
so
that makes sense. That makes perfect
does it
sense.
though? If it should be measuring something about aging that's relevant to mortality, shouldn't it? I mean, I know it's not trained on that, but what is it? I'm trying to interpret what that means.
Well, it does. I presume it does marginally improve upon using calendar age. You say it does not. Well, yeah, that's interesting if it doesn't, because that would contradict the. I mean, so the analysis done on the Lothian birth cohort data as well as the Generation Scotland data does show that even so the Horvath clock, for instance, does actually correlate with all-cause mortality, but not as strongly as the second generation.
Yeah, which is what I expected to see. So.
Yeah. So it's surprising if it did not.
So if you train a clock,
yeah, right. Yeah,
so
I'm not.
you train these clocks now to predict chronologic age within a year or so, and biologic age almost certainly has to vary more than that. So I'm just wondering, they're overfit, but I'm wondering what they're actually measuring. So it's
in terms of the actual processes, which are the processes that are sort of well, I think that so what we showed in our Nature Aging paper was that the more accurate a clock is for chronological age, this includes the first generation clocks, that the more of the actual process or methylation changes are going to be stochastic. So, and that makes sense because that makes sense because it's counterintuitive, but actually think about, remember that you're measuring a cell population. So if you have a purely stochastic process at the single cell level, because that's where the stochasticity is, at the single cell level, then at the cell population level, by the central limit theorem, this becomes a very tight distribution. So you actually get a very accurate predictor for chronological age or time, basically. So it makes sense that the more stochastic is. So this is going to be, I mean, these are stochastic changes that are related in part. Some of it is going to be cell division, but of course we know it's not just cell division because it also predicts in post-mitotic tissues. It could be related to the somatic mutations that Trey Adeka has been talking about, the spontaneous deamination of cytosine to thymine. These somatic mutation clocks, by the way, as seen in cancer, they also predict chronological age really well. So that could be it.
Anyway, I don't want to take up the whole seminar. We can have this. We can continue this over a beer. I'm just really curious how what we're actually measuring.
What it's actually measuring.
Yeah. So I want to bring in Lim Jiming though to ask some questions from the audience.
The main question was like are aging clocks are ready to be used in clinical practice, and also why do we have so many clocks out there? So if you want to add some points, especially for this audience, they are more laymen, you can try to answer this question to make them understand. And also, I think they can bring take-home message like, since there are so many clocks out there, I think it's too blunt to ask you go and suggest them which company to use. Maybe you can give them some ideas like if those people they really care about their biological clocks, how they're going to choose it wisely, and what question they're going to ask the company, or look at a catalog. Are you adjusted for your cell composition, something like that? Yeah, give some suggestion to them. Yeah.
Right. So basically, I sort of gleaned two broad questions here. One was the technology. What technology should we use in the clinical application? And then the second question is which clocks, because there's so many clocks out there. So let me address the second question first, because I think that from my perspective, I think that, and this usually is what happens, right? So too many rush. There's a rush to to bring this into the clinic and to make this translation. And indeed, for instance, I've actually heard about this at the geophysics meeting, but there was also a Nature article about this, that a company like True Diagnostics, they were having trouble with the reliability, right? So an individual person might get very different measurements for 2 samples taken 2 weeks apart. And actually, I think that one big source of variability that explains this heterogeneity is precisely cell type heterogeneity, because the immune cell composition of a person does vary quite a lot as a result of many factors. So, I mean, were you jet lagged when you gave the sample? What was your diet the day beforehand? All of this is going to affect your immune cell composition. And if you're not careful in how you build the clock, you're going to have this source of variability. So, so we actually saw associations. When we did this meta-analysis, we saw associations of immune cell fractions with many factors, including exercise, including sleep, hours of sleep, caffeine consumption, etc. So all of this is going to affect, and so you need to be more careful about how you build the clocks. So I think it's not a question. I mean, I don't think that any of the current clocks really can be applied, because, as I mentioned before, they don't give you a confidence interval. They don't tell you what is the probability that you're really aging unhealthily. I mean, what does a value of plus 0.5 really mean? Is it statistically significantly different from 0? Nobody is addressing that question. So you need to address that first. Otherwise, you're just, unfortunately, just making money out of the customer, I think. So as far as the first question is concerned, which technology? No, I don't think you would want to use, why would you use array-based? I mean, a customized array technology, probably yes. If you once you've honed in on a number of CPGs. So I do think that for robustness reasons, you will want to have on the order of 100 CPGs. So you want to have redundancy. Redundancy in a machine learning predictor is a good thing. It's not a bad thing. It gives you robustness. So for instance, our syntactic convolution algorithms, their robustness relies on having redundancy. We don't just pick one CPG for each cell type. You want to have 50 or so, because that gives you a really strong, robust estimate. And the same thing applies to these clocks. And so a customized array would be a way forward, I think, because the reliability of these arrays are quite high. I mean, bear in mind that you can detect a 1% change in methylation with 95% sensitivity. And this is clearly demonstrated, for instance, with smoking-associated DMCs. These are really small 1 to 2% changes. And actually, if you take all the smoking US studies, the top-ranked CPGs are very, very stable. I mean, they keep the ranking is quite stable, which suggests that there is that the assay is quite accurate, right? But then again, there are other technologies as well that could be used.
Some changes in DNA mutation with age possibly reflect a failure of maintenance by the epigenetic machinery. What is your opinion on shifting the focus away from DMPs or VMPs and look into stable CPGs to gain insights on protective mechanisms on the epigenome?
Right. Yes. Well, I think if I interpreted this question correctly, this relates to this taxonomy I was showing, this age-related taxonomy. So it's intriguing. There are these CPGs that are very stable, except for a small number of outliers. Yeah. And the big challenge there, though, is that some of these outliers are technical. That's clear. We've got evidence for that. Okay, but not all of them are, and being able to disentangle the two could be challenging. But I agree that this is it's certainly an interesting avenue to explore, because if they are somehow homeostatically vital, then suddenly you've got in one particular individual, you've got this big change in methylation, then this could be important, but could have an important effect biologically, because why is there stability at these loci? There must be a biological reason why there is this stability, right? So if you break that stability, then this could be important. So I think that it would be interesting to have matched methylation and transcriptomic and proteomic data to actually explore that hypothesis.
I have to jump in because we're out of time. Thanks, Andrew, for doing this. Great discussion. We need to have a beer together. Thanks.
Well, let me know when you're coming to Shanghai. Then we can do that, or I'll try to come to Singapore.
Sounds good, one way or the other. Thanks a lot. Thanks, Jimmy. And I want to remind listeners to use the chat function and panelists and all attendees option to leave comments and feedback on the show. Next week, we have Tony Weiss-Corey. That'll be the 15th, and he'll be talking about young blood for old brains. And then before we close, I want to know if anyone still remembers Ralph Macchio. If you don't remember him, he was the Karate Kid from the original movie. He's now in his 60s, and I bet we all want to know what he looks like now. Thanks for joining us on the show.
Why does Ralph Macchio look so young? It's my parents' fault. It's good genes, it's youthful boyishness that I lean into.
At what age as a kid did you realize you weren't getting older?
It's always been that way.
How old are you?
I'm 18.
That's hard to believe.
I know.
Do you mind me asking how old you actually are?
No, I don't mind you asking how old. Shall I tell you? I've just turned 30. So that's the amazing thing. You know, someone asked me, is it traumatic turning 30? But for me, I think it's traumatic when I look 30. So, so I figure another 20 years I'll be there, and then I'll deal with that.
Do people lose their minds when they find out your age?
Lose their minds?
Yeah.
No, maybe a little surprised.
Because you look almost exactly the same as you did, and yet... Do you mind saying how old you are?
Do I mind? No, I'm 60.
60 years old.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.